So, good afternoon here from Spain. It's my pleasure to introduce Dr. Matt Daugherty, a Cooperative Extension Specialist in the Department of Entomology at the University of California, Riverside. Matt holds a bachelor from UC Davis, a master from Illinois State University, and a Ph.D. from UC Berkeley. His research and extension work is on the ecology and management of invasive insects and vector-borne plant diseases. His recent projects have explored the resurgence of pest disease in Northern California vineyards, the invasion dynamics of Asian citrus psyllid, and the risk assessment of a moth pest complex affecting California vineyards. With that, I turn it over to Matt to begin his presentation.
Great. Thank you, Rocio. Appreciate it. And thanks also to Blanca and to Leo for the chance to talk to the group. So let me share my slides. Can you guys see that?
Yes.
We're good? Okay, great. Thanks. So what I was going to do today is just to warn you, this will be not so much about Xylella or transmission or anything that I've worked with Rodrigo on for many years. Instead, I'll mostly focus today on some recent work we've been doing trying to understand a little bit better now 20 plus years into the glassy-winged sharpshooter program. What kind of evidence do we have that some of the regulatory programs that have been put in place are actually having the effect that we want them to have? So that's what I'll do today is go through the evidence that we have, particularly related to some of the nursery quarantine work here in California, to make sure that glassy-wing is not continuing to spread any further than it already has in California. And please, I'm happy to take questions. Please jump in if something doesn't make sense. I'm happy to take them all. You don't have to wait to the end. So up front, though, I'll acknowledge folks whose background work really set up a lot of what I'll talk about today, namely Rick Reddick, Jim Bethke, and John Cabashim, a couple of farm advisors, and then at least a couple of people at the CDFA, notably Stacy Oswalt. And Stacy's sort of the gatekeeper for a lot of the California Department of Food and Ag program data, especially that related to the nursery program. So thank you very much to Stacy for sharing all the numbers and the data that I'll present to you guys today. All right, so the context here in California, as you probably all know, is we have a really interesting but also pretty complicated complex of vectors that play a role not only in Pierce disease, but also other Xylella diseases. And so it's complicated because it's really dynamic, both spatially and temporally, in terms of which vector or vectors are really the most important. It can vary a lot, location to location, also among cropping systems, things like that. And so it has been hard to get through to understand the complete picture here. And we actually continue to try to reevaluate some of this information to make sure we have the details correct. So a lot of it has been driven here in California historically by research and our understanding of what we know about many of the native vectors. So a lot of that work has been focused in the Coast Range, specifically on the native blue-green sharpshooter, which is a really efficient vector of Xylella to grapes. There are at least a couple of other largely grass-feeding native sharpshooters, things like the green sharpshooter and red-headed sharpshooter, that can be locally abundant, particularly on the edges or in pockets of vineyards. They can also play a role in other cropping systems like alfalfa. And so specifically the green sharpshooter probably is an important vector of Xylella in alfalfa. We've also known for a long time, and in recent years started to make sure we appreciate how much spittlebugs might be playing a role. This is all in light of what you guys are facing in Europe, have seen over the last decade plus. And so a lot of work recently, most of it spearheaded by the Almeida Lab, is trying to understand what role some of these spittlebugs, particularly meadow spittlebug and at least one Afrophorus species, might be playing in our PD systems in California. And then we have at least a couple of other vectors, native vectors in California as well, that may play an important role, at least in some locations. And so willow sharpshooter is one, and I mentioned that because it's speculated that might have been the primary vector down near where I am in Southern California, historically prior to the arrival of glassy-winged sharpshooter. It's sort of a riparian species and can be problematic in some areas, really throughout the state and the Coast Range. And then the other native we have, particularly in some of the interior sort of desert regions in Southern California, is this congener of the glass-winged sharpshooter known as the smoke tree sharpshooter, Homolodisca lacerta. Not especially efficient and really not all that abundant, at least in most of the places that we grow grapes in California, but in a handful of cases it is out there. And really what I'll focus on today and what has really completely shifted and been the focus and shifted all of our efforts in California over the last 20 plus years has been the invasion of this new vector, the glass-winged sharpshooter. And this is an insect that's native to the southeastern U.S., so Florida and the Gulf Coast. But it's been here in California. At least it was first reported in the late 1980s from some residential areas in Southern California. We honestly don't know how it arrived here first. It's really the source of speculation, but regardless, it's been here for 30-something years. And in that first decade or so, it wasn't all that abundant, but then eventually started to really take off. Its populations really ballooned through the 90s or the 1990s. And so by the end of the 1990s, its populations were extremely high, much higher than we typically see for the native vector populations. And then we started to see all these novel Xylella diseases that hadn't been noticed before, things like oleander leaf scorch, and then some scorch and many of the other ornamentals, especially in Southern California. Things like liquid amber trees were showing leaf scorch symptoms, jacarandas, some of the ornamental plums, all showing evidence of these new Xylella diseases that hadn't been noticed before. But then ultimately, by the late 1990s, glasswing's super high abundance really triggered what's probably the most clearly best documented severe epidemic of P.D. in California, and that's this epidemic that occurred in Temecula Valley near where I am, starting in the late 1990s. And I'm sure you guys know the details of this, quite devastating to the industry down there, with estimates of more than a third of the vines, maybe 40% of the vines lost to Pierce's disease in just a few years, 3 or 4 years. And then it also started to follow suit in parts of the Central Valley, as I'll show you in a little bit. Parts of Southern Central Valley started to see similar issues once glasswing populations really increased there. So one of the things I'll come back to in a bit is, and it's become clear, is the impact of glasswing. This is work that Rodrigo and I have done over many years at this point. It's not that glasswing sharpshooter is all that efficient a vector compared to some of the natives, especially blue-green sharpshooter. Its impact appears to largely be driven by the fact that it can achieve way, way higher densities than our natives do. And in most of California, that's driven by the fact that it's able to take advantage of citrus groves and significant commercial citrus acreage, because citrus trees are really great overwintering and reproductive habitat for glasswing. And so we see big problems historically in vineyards that are adjacent to commercial citrus production, which is in the southern part of the state. And again, we'll come back to kind of why that matters in a bit. So this map here is our understanding of where glasswing sharpshooter's current range is, 30-something years in at this point. And so these blue regions are where it's considered to be widespread and quite abundant in most cases. And that includes most of the Southern California region, and then at least a few counties in what we refer to as the Southern Central Valley or San Joaquin Valley. That includes part of Kern County, a little bit of Tulare County and Fresno. And in most of these areas, that little finger of blue you're seeing there where glasswing is established is in and around where there's lots of commercial citrus planted. And it's been pretty stable, its distribution in these blue regions for probably 20 plus years at this point. I'll show you a handful of exceptions to that in a minute, but generally it's been here a long time, but it's mostly been in these blue regions and been that way for quite a while. Nonetheless, there's a lot of concern that it could invade other parts of California, including all these green counties, which are considered at risk to either glasswing establishment and or Pierce's disease. But currently, with some exceptions, it currently is not known to be widely established in any of these regions, including throughout the really premium wine-growing regions in the coast of California and the North Coast, as well as the northern parts of the state. So that's the concern, and has been the concern for a long time, is to make sure that that does not happen, that it does not continue to spread beyond where it's currently established. And there's a reason to believe that if it did, it could cause serious problems. And it's in part based on the fact that PD already is a really significant economic issue in California. And this is based on analysis by some UC Davis economists who estimated, and this is about 10 years old. They're currently working on updating these estimates, but as of about 10 years ago, they figured that Pierce disease, the direct and indirect costs of Pierce disease to California was about $100 million a year in a quote unquote average year. And some of that's direct losses due to Pierce's disease, losses of vines. A lot of it, though, is increased management costs, increased resources devoted to research, and then a lot of the regulatory programs, both in terms of nursery as well as area-wide programs, most of which is focused on containing or improving management and control of glassy-winged sharpshooter or Pierce disease associated with glassy-winged sharpshooter. So $100 million a year, but the main point is that those economic costs or losses are not distributed universally across the state, of course. And in fact, currently, they estimate that the majority of the costs, if you read the fine print in this analysis, the majority of the costs are actually occurring in areas outside of where glassy-wing is already present. And so just Napa and Sonoma alone appear these green regions in the map. They estimate that somewhere between 50 and 90% of the vine losses due to PD, the economic costs of those losses are accrued in those areas. And that's because those areas are incredibly valuable property with incredibly valuable high value yields from those regions that kind of dwarf some of the losses in other regions. So that's a long way of saying that a lot of the economic cost right now to grape growers is not in areas where glassy-wing is. And so that begs the question of what happens if it does spread into those areas. How much worse is it going to get? So that's the concern. And that's been the concern for a long time because it seems quite plausible that glassy-wing could become a lot more widely established than it initially was, or even than it currently is. And part of the reason for that concern is that glassy-wing is pretty notorious in terms of being an extreme generalist. It uses plants to varying degrees for feeding and or reproduction. Upwards of a 300 plant taxa have been documented to be used by glassy-wing. It unfortunately includes a bunch of agricultural crops, not just citrus and grapes, other things as well. And also like dozens, or if not hundreds, of some of the most common ornamental plants we plant in residential areas of California are ideal for glassy-wing. It really likes them. And then to a lesser degree, some of our native species, especially some of our native trees and woody shrubs, are used a bit by glassy-wing. So there's a super broad host range, and that presumably means that there's a decent risk that if it's introduced into an area, it could probably establish to some degree because there's almost surely going to be a plant there that it's able to take advantage of. It's not even close to a specialist. And that makes it kind of hard to predict where exactly it might move next is part of the concern. Although there's a caveat to that, and I'll talk about in a second. The other piece of that concern of it spreading more widely, it goes back to what I talked about, here with when it readily uses dozens or hundreds of common ornamentals. Unfortunately, a lot of the production of those ornamental species right now is occurring in Southern California in these blue counties where glassy-winged sharpshooter is already established. And so this is Orange County, Los Angeles County, Ventura, San Diego, and then near where I am in Riverside County are the dominant producers of nursery stock, ornamental nursery stock that then can move more broadly outside of those environments. And so the CDFA, California Department of Food and Ag, has records of the shipments of plant material out of these regions, and they estimate since 2001 approximately 1.2 million individual shipments of plants coming from these glassy-wing infested areas to outlying areas. And that's a total of somewhere on the order of 300 million plants in the last 25 years have been shipped out of these infested areas. And so obviously, if there's no oversight, if those plants are not protected from glassy-wing, there's a really substantial potential for them to be the movement of that nursery stock to contribute to the unintended spread of glassy-wing. And that's been the concern for 20 years. Unfortunately, as a result of that, there's been pretty stringent restrictions placed on the nursery industry to make sure that risk is reduced somewhat. And again, what I'll talk about today is how effective does it seem like that actually is. Okay, so quickly, the nursery program, there's a couple of variations on what's required of nurseries these days. And so the sort of traditional version of the nursery program, so for any of these nurseries in the southern part of California where glassy-wing is established, they're required to enter into what we call a compliance agreement to ship plant material out into outlying areas where glasswing isn't present. And they have to be free of all sharpshooter life stages in order for the shipment to go through. That requires that the nursery do some background monitoring in and around their production facilities. So they have to deploy these sticky traps at a set density and then check them on a regular basis. And if they get above a certain number, I think it's something on the order of like 10 sharpshooter adults in the last month or two, a temporary hold is placed on the movement of the plant material out of the nursery, and they have to do an insecticide treatment to knock down the glasswing populations before they can start shipping again. So there's this background monitoring and treatments that have to occur in these nurseries. The other thing that they're required to do is they have to do these really time-consuming and costly visual inspections pre-shipment of the plant. So they have to visually inspect the entirety of all of those glasswing sharpshooter host plants that they're going to ship out to make sure that there's no life stages present. And then typically there'll be an inspection on the other end wherever it gets shipped, typically by either the county or by the California Department of Food and Ag, to make sure that the shipped material did not have any sharpshooters on it. So that is really time consuming, both for the nurseries as well as for either the counties or for the CDFA to do these things. And it's especially doubly costly for the nurseries because if a shipment gets found with any glasswing on it, it's either rejected and not allowed to be shipped out and or has to be destroyed. So they theoretically lose a lot of plant material on top of already the costly labor involved with doing these inspections. So that's been a problem and estimated to be well over $10 million a year to the industry. So a little bit more recently, the CDFA and the Peer Disease Control Program offered a slight more flexibility to the program where they said you can drop that pre-inspection requirement. So you no longer have to visually inspect the plants before they go out. But instead, you actually have to do one of these mandatory prophylactic disinfestation treatments. And so what they're required to do is basically set aside the plants that are going to ship into a certified glasswing-free zone temporarily, apply one of these two products, a carbamate, referred to as tame, or a pyrethroid known as Savin. These are both approved for glasswing use in nurseries. All the plants have to be treated ahead of time with one of those two products and then loaded up quickly onto the truck within 5 days and then shipped out just to make sure that they're protected adequately. And these two products are the only ones approved as part of this, what I'll refer to as the ATP or approved treatment program. And that's based on some work by one of my colleagues, Rick Reddick. They did years of work evaluating different insecticides on nursery stock and found that these two do a really good job, upwards of 95 to 100% mortality of glasswing sharpshooter egg masses on nursery stock. And as I'll show you in a second, it's the egg masses that are most likely to be found on nursery stock. And so these two products are the only ones that are approved just because they're so effective at controlling and killing glasswing sharpshooter egg masses. So those are the two programs that have been in place here in California. One, so the basic version of this nursery quarantine for more than 20 years, and then this slightly newer version, this ATP, since about 2009. So what I want to do now is walk you guys through and talk a little bit about what evidence do we have that this nursery program has had its intended effect of limiting the spread of glasswing. And so I'll quickly kind of give you an update on where things stand in terms of glasswing, kind of where it's been found since it arrived, and then kind of what the response was and what the outcome is. And then I'll get into analysis of some of the program data that the CDFA has collected on the number of shipments going out, how often they're being intercepted with glasswing, things like that. And then I'll quickly touch on a bit of interesting biocontrol data where they're actually bagging some of those egg masses that are found and estimating the amount of biocontrol, which turns out to not be trivially small. It's actually maybe a little bit more important than we think. So I guess quickly though, before I get into the actual analysis, which I'll spend the rest of the time, is there any questions about stuff up to this point, maybe particularly about any details on the nursery side of things? No? Okay, I'll keep going then. And again, jump in if you have any specific questions on anything. Okay, so here's where things stand, and I'll just quickly go through this again. But this is where things stand in terms of glasswing, where it's basically been found over the last 20 plus years. And I'm not showing you anymore, but again, remember that it's considered widespread and established in these blue regions in the southern part of the state. what I'm showing you in these stars here are what I'll refer to as incipient invasions, and these are localized detections of glassy-winged sharpshooter. And there's been 21 of them since 2000, so less than one a year, basically on average. And so these detections are occurring because there's essentially a statewide monitoring program using sticky traps being deployed in all of these at-risk counties in green that are checked on a regular basis to try to identify if glasswing is introduced into an area, just to capture it as soon as possible. And so those 21 stars are locations where this has occurred. And so what happens is if there's a detection that triggers additional monitoring and inspection of plant material in the surrounding neighborhood, and it also triggers insecticide applications of in the surrounding landscape, especially if live stages of glasswing are found. And then also releases of some of the biocontrol agents will also occur in the surrounding areas just to make sure that there's effective IPM in and around these incipient invasions. And so of those 21 that have occurred so far, 19 of them have been successfully eradicated to date. And those are all the red stars here. So there's a handful of them in the early days down in the southern part of the state, Imperial County. There's one in Fresno early on nearby where now glasswing is actually established. But then the bulk of these other detections have occurred in coastal areas and locations in the northern part of the state, as far north up here as Butte County. But 19 have actually been eradicated. The two that haven't so far, the two newest detections, that's in Solano County, which has been going on for a few years. It's one of the more kind of persistent ones. And then the most recent within the last year is on the edge of El Dorado County, right near the city of Sacramento. And so we'll see what happens with those two. But by and large, not only has it proven possible to detect these things early enough and actually success eradication, but there's also no indication, to my knowledge, that there's been any widespread, any spread beyond the initial detection locations for any of these incipient invasions. So it appears that this program has done a really good job at detecting glasswing pretty early on, and there's a successful strategy to eradicate them once it is found. And so we'll see what happens with these two most recent ones, but arguably this is pretty compelling evidence that we are able to, at least if there's a limited number of these introduction events, we are able to deal with them pretty successfully. And I won't go through this too quickly. A lot of that just appears to be just a really effective program that's in place. There's probably an argument to be made also just in terms of where many of these introductions are occurring, that we're also getting a little bit of an assist because a lot of these northern locations may not be ideal based on what we know about the thermal performance of glasswing. They're being introduced into areas which aren't as ideal as the areas of Southern California where it's widespread. And that's based on some demographic modeling by some of my colleagues, including Mark Hoddle. And the other piece of that here, I think it's also not a coincidence that we're able to successfully eradicate and they're not spreading more widely and achieving really high densities in many of these locations because they're in areas where there's not a whole lot of commercial citrus. And so that's what's being plotted here is this orange points here is the distribution of commercial citrus in California. You can see a lot of it's in Southern California, and then that portion of the Central Valley up to about Fresno, which is the range of where we consider glasswing to be widespread. Most of these other locations outside of the San Luis Obispo detection here, most of those northern locations, there's not a whole lot of commercial citrus. So that probably is also helpful in terms of our ability, the CDFA's ability to successfully respond to those incipient invasions. So that's kind of where things stand. And again, this long way of saying that, yeah, glasswing has occasionally been introduced into other areas of California. So far, it has not actually caused problems in those environments. We've been able to contain them. And so its invasion front has really been pretty stable in the southern half of the state for more than 20 years. Okay. And so the last piece of that, and what I'll spend the rest of the time going into is why haven't there been more of those introduction events beyond the 20 or so in outlying areas? Why hasn't it happened more frequently? And that's where the nursery quarantine program comes in place. And so I'll walk you through two sets of analyses, as I mentioned. One is all this great data that the CDFA has collected as part of these compliance agreements with nurseries. They have a really nice idea of how many shipments are going out and to where and how often glasswing are being found on those. And then I'll talk again briefly about a little bit of data where they're tracking the level of parasitism on some of those egg masses that are being intercepted. Okay, so let's start with the shipment data and the number of interceptions. So I mentioned that since 2001, I guess, there's been roughly 1.2 million unique shipments out of these areas that are where glasswing sharpshooter is established in the southern part of the state. 166,000 of those 1.2 is part of that ATP, the approved treatment program, where they don't have to do the inspection, right? They can do the prophylactic treatments. Of those total of 1.2 million shipments, there's been a total of 639 interceptions. Only 2 of those are coming from the 166,000 ATP shipments. So across those 2 sets of quarantine programs, right, we're talking about 99.95% of the entirety of these shipments, of these 300 million plants, appear to be uninfested. So that amounts to 1 interception per roughly 1,900 shipments. That, just at face value, seems like a pretty effective program, right? Seems to really be capturing the vast majority of the risk associated with the movement of this nursery stock. Nonetheless, it looks like there might be a difference between the standard non-ATP program, the historic program, and this newer ATP program. So that's what we wanted to analyze, just to start with that, is to see if there's a significant difference between those. And so to do that, we used a logistic regression analysis with what's referred to as a birth bias correction. And that's just to capture the fact that these are really, really rare events, especially for detections of the ATP. And so we need to kind of correct for the rarity there to get a better fit for the model. And so in this analysis, we're simply comparing the program type, either ATP or the traditional quarantine program, as a fixed effect. And then we're also including year in there as a covariate to capture, to see whether or not the rate of interceptions is actually changing over time. And the results are incredibly clear-cut. So there's a really significant effect of nursery program type difference between the ATP and the standard non-ATP, and that's shown here. It's a little hard to see here, but basically the frequency of ATP interceptions is 50 times lower than the normal program. So that's a difference of 1 interception per 1,600 shipments versus 1 per just 83,000, at least based on small sample sizes. So that's, again, the overall success rate for the program is 99.95% of the shipments are going out clean. For the ATP program, it's actually closer to like 99.99%. So really staggeringly effective program as far as we can tell, based on the shipment data. So there's a clear effect, and the ATP seems to do a better job than the visual inspections, which is potentially useful information. The other part about it, which is a little bit interesting to me, is that there's a really significant effect of year as well on the frequency of glasswing interceptions on nursery stock. And you can see that there's a striking decline, right? A really sharp decline over time, where interceptions are relatively uncommon, 0.25% of shipments early on. But now, over the last, 15 years or so, it's down under 0.01% of the shipments. So again, not only is this quarantine program effective, if anything, the last 15 years, it's become even more effective. And there's questions about why. I can't really answer with certainty. My best guess is why it's been more effective the last 15 years is probably because glasswing populations have declined over time. We have some rough evidence from that, not so much in residential areas of Southern California or suburban areas where these nursery production is occurring. But that would be my best guess, is populations the last 15 years in parts of Southern California is lower than certainly what they were in the early 2000s. Regardless, the program, again, is becoming, if anything, has been more successful over time, which is good news. So that's the difference between the ATP and the regular quarantine program. But we want to dig into that a little bit more and look at other sources of variation in interception rate. And we can do that with this data set, fortunately, at least a subset of the data. We can look at differences among the county of origin in terms of where these shipments were coming from, as well as the seasonality in that, and also different glasswing sharpshooter life stages that are detected. And so this reduced data set has 620 total interceptions. 92+% of those are egg masses of glasswing being found. So it's pretty rare to find nymphs or adults. Most of it is the egg masses potentially being shipped on nursery stock. And so that's what we'll focus on here with this next set of analysis, because we just don't really have enough adult or nymph captures to do anything with the analysis. So we looked at source of variation and interception rate in the egg masses of glasswing with a binomial generalized linear mixed effect model, similar kind of model that includes a fixed effect of year and month, again, to capture some of those temporal effects, and then a random effect of which county the nursery was located and where it shipped from. Okay, and so that the temporal effects are shown here. And so on the left, I'm again showing you essentially the same significant effect of year where we see this sharp decline over time. So it just becomes rarer and rarer and more recent in the last 15 years to find sharpshooter egg masses on nursery stock, let alone the nymphs and adults, which are almost never being found. So that again matches up, and again we can't say for sure, but again my best guess is that it's because populations over the last 15 years have been lower in Southern California than what they were historically. The other part, though, is the seasonality, the time of year they're being found. And that's shown in the right-hand graph. So we'll focus on the white diamonds here, which are the egg masses again. You can see that there is significant difference over the year in terms of how likely you are to detect an egg mass, intercept an egg mass on nursery stock. It's really rare overall, but it's a little bit more frequent at certain times of year. So we see a little bit of a peak in the spring, February, March, April. That drops down, becomes uncommon again May, June, and then we see this summer peak in July and especially in August before it declines again. So the likelihood of detection is not the same over the year. That indicates there's differences in the seasonality, the activity of glasswing and egg laying on nursery stock. And fortunately, there's two useful things about this. First off, this seasonal pattern more or less matches with what we know about the activity of glasswing in commercial citrus and in vineyards. So that's reassuring. It's kind of doing the same thing in nurseries. And the reason that might actually be somewhat useful is if you wanted to further optimize the program based on this, you would say, okay, maybe July and August, if there's some extra special steps we could take, maybe we implement them at that time of year to further reduce the chance of shipments going out infested. So yeah, the point is that there's seasonality. Maybe we could use that information some way in the future. Okay, so there's temporal effects that are driving glasswing infestation rates on nursery stock. The other part about it is that what I'm showing you here is that this is the random effect of the shipment county where the nursery was located. It's broken down by the 7 Southern California counties where we have shipment data for. So the numbers above the bars is the total number of shipments since the program was started by county. You see some ship a lot more plants than others. But then the bars themselves are showing the proportion or percentage of the shipments where their interceptions occurred. And you can see that there's a lot of variation there. And so unfortunately, my county, Riverside and Ventura, have higher interception rates than some of the other ones. And I'm not pointing fingers and blaming people or counties, but Ventura and Riverside are a little bit more problematic, I would say, than other places like, say, Orange County or Santa Barbara, which have something like 10 times fewer lower frequency of interceptions. And again, I can't really say for sure what explains this. There appears to be some at the county level, some differences probably in the abundance of glasswing. We don't really have the monitoring data independently to say for sure. But the other part about that is it's maybe not a coincidence that Riverside and Ventura have the highest interception rates at nurseries. Those are two of the counties with essentially the biggest commercial citrus production as well. So it probably is feeding into the infestations in nursery stock. At least that's my best guess. Anyway, so there's spatial variation in addition to temporal variation going on here in terms of the risk of nursery stock being infested. So again, hopefully that's somewhat useful information as well for the regulatory program. All right, and so then I'll finish up with a brief analysis of a little bit of data that has come in related to biocontrol of glasswing sharpshooter egg masses. And it turns out that glasswing are fortunately attacked by a range of beneficial insects, including predators, and their egg masses in particular are parasitized by several species of wasps. Most of them are in the family Mimeridae, and those are in the genus Cosmocommodia. This used to be known as Gonatoceras, so I think that's what Cosmocommodia ashmeadi, I think, on the top, and I maybe moralized as the middle picture. And this is an egg mass, a parasitized egg mass on the bottom. So there's probably what 6 or 7 parasitoid species that are known to attack glass-winged sharpshooter egg masses. They were actually mass reared and released by the CDFA for many years. They've now kind of limited rearing to, I think, just a couple of those species, and those are still occurring in the northern part of the state, in the Central Valley, and around those new detections of glass-winged sharpshooter. They'll go in and do releases of some of these parasitoids that they're releasing. The Southern California piece of that program was halted in 2012. My understanding is due to just financial limitations. Um, so it turns out that the nursery, as part of the nursery program, and when they're intercepting these egg masses, they're actually bagging a subset of those egg masses and looking at parasitism, parasitoid emergence from those. This is kind of useful because it allows us to evaluate what kind of biocontrol might be going on in these nursery settings, which I think it's safe to say we oftentimes assume is unimportant generally in terms of IPM of pests. And we'd like to be able to evaluate whether that's really a safe assumption or not, whether biocontrols may be having a little bit of a bigger impact. And as I'll show you, I think it might be, which is cool. So since 2008, there's been 102 egg masses as found as part of inspections that are bagged, and then parasitism rates were tracked on that. This occurred over most of the year between February and November, so we have at least a bit of seasonality data on that, and it's on 37 unique plant taxa. There's been evidence, or there's been egg masses pulled from. So we look at parasitism rates among different plant taxa. And so we again analyze this with the generalized linear mixed effects model with binomial error, in this case with a random effect of the different types of plants that they were found on. Okay, so overall, the data we have, it sounds like they didn't keep track of the number of individual eggs per egg mass that were parasitized, or then they also didn't have the ability to identify the parasitoid species. So we don't have that information, but we do have information on just whether there was any evidence of parasitism of the entirety egg mass at all or not. And it looks like not quite a quarter of them. So 22.5% of those 102 egg masses showed at least some level of parasitism, which is cool. It's really actually not. It's higher than I would have expected it to be, let's say. And again, we don't know which parasitoid species are responsible for that. There's a couple of likely culprits, just because there's a couple of species that appear to be the most important in terms of their impacts on glasswing. So we probably have a rough idea, but something that would be nice to follow up on. And then of those 37 different nursery taxa where their egg masses pulled from, 13 of them showed evidence of parasitism. It turns out to be a wide range, if you recognize these plant genus genera. It's a wide range of ornamental species, including some native, some fruit trees, and then just some more ornamental species. Okay, so let's talk about some of the other sources of variation in parasitism rate. So one of the things that became apparent, I need to follow up with the CDFA. I'm not sure I fully understand what this is showing, but it appears to be, I think after an egg mass is found, I think it was those plants were treated with one of the two ATP products, either 7 or tame. And then they bagged the egg mass and looked at parasitism rates. So I may have the details of that wrong, but the main point is that parasitism rate differed between plants that have been treated with 7 versus tame. And specifically, tame looks like parasitism rate, it induces or allows about 2 times higher parasitism rates than 7 does. And this is somewhat interesting because assuming I'm interpreting this right, it suggests that maybe one of these products is a little bit more compatible with biocontrol than the other. It's something that would be worth following up. So perhaps a little bit of difference based on which insecticides are used. There's also a really strong effect of seasonality or month of the year on parasitism rate. That's being shown here. I should point out that this November point here is a little misleading because it's based on, I think, on just a couple of egg masses that are found in November, both of which are parasitized. So it sort of exaggerates the effect. But even if you ignore that, there's a significant increase in parasitism rate over time. That is good to see because it very much matches up with what we know about parasitism rate in, again, commercial citrus and vineyards. Especially a couple of these parasitoid species take a while to get going. Their activity is very low in the winter and spring, and they really don't get going until late spring through summer. And we see that here. We can see this increase in parasitism from roughly May through the end of summer. So that matches up with what we already know, which is great. And again, just highlighting the fact that there is some parasitism occurring in these nursery environments. And then this is maybe the more surprising result to me, and this is showing a significant effect of year on parasitism rate. And again, this point in, I think it's 2023, is a little misleading because it's, again, those two egg masses that were found in November, both of which are parasitized. But ignoring that point, we still see this significant increase in parasitism rate over time. And I don't have a great explanation for this. It's very interesting to me, specifically that since the mass rearing and releasing program in Southern California was halted, we still see this increase in parasitism rates over in nurseries, to the point that in the last few years, since 2020 or so, the majority of the egg masses being found in nurseries are parasitized. That's pretty substantial biocontrol going on. So perhaps further reducing any risk that the handful of egg masses going out that do get through, looks like a fair number of them might actually be attacked by these parasitoids. So further reducing the risk of moving infested nursery stock. So this is something I'd love to follow up on a little more to know how real this temporal pattern is and what explains it. And then the final result I'll show you is that random effect of parasitism rate based on the plant taxa that the egg mass was on. And so of those, 37 taxa, parasitism was found on 13 of them. And then showing you that here. So in several of those plants taxa, there's no evidence of parasitism. But then in some, it was quite common. And so 3 of these that I'm showing you pictures of. So Metrosidera, which is this bottle brush plant, which I think is native to New Zealand. And there's only a handful of egg masses, but all of them were parasitized. Podocarpus and Tristania, a couple of other ornamentals native to other parts, I think parts of Asia, I believe, were also the majority of the egg masses on those were parasitized. And so it suggests there's a lot of variability here. It's a little too early to tell because there's not a ton of data, but it suggests there might be some sort of interesting host plant dependency in terms of the activity of parasitism. So it'd be nice to kind of follow this up a little bit to know how much variability there and how much the plant itself might be influencing parasitoid activity. So I'll wrap up there. The main take-home point of this, if you got nothing else out of it, is hopefully I've convinced you that glasswing has not really significantly increased its invasive range over the last 20 plus years since this regulatory program has been put in place. So we've been really, and I say we, the royal we, have been very successful at limiting the expansion of glasswing, which is great. Part of that appears to be due to that early detection eradication program. It looks like those efforts are quite effective, at least to date. 19 of those 21 eradications have been successful. As I mentioned briefly, I think part of it also, we might be getting a little bit lucky in that some of the introductions are occurring in environments which really aren't ideal for glasswing because of either climatic conditions and or lack of commercial citrus access. So that's part of it in terms of explaining why it hasn't expanded its range more. Sorry, I got a phone call. The other piece of it is relates to the nursery part of it itself, though, and how it appears to have been really effective at reducing the unintended risk of.
You reached the Daugherty lab. Please leave a message and we'll call you back. Max,
sorry, guys.
directly on the cell phone.
There we go. So as I try to show you, the vast majority of the nursery stock moving out is uninfested. Most of the sharpshooters are being captured by that quarantine program that's been in place for at least a couple of decades. The approved treatment program, this newer version of it, seems to, if anything, do an even better job at reducing the chance of nursery stock moving around infested. And then the final piece there is that it looks like biocontrol is maybe having a little bit more of a role than we expected in these nurseries. And it's something we'd like to follow up with a little bit more. So I'll wrap up there. And thanks again to the folks who made this work possible, especially Stacy, for sharing all the data that I presented to you guys. And I'd be happy to take any questions if there are any.
Okay, thank you, Matt, for sharing this insightful presentation. So now we will love to hear from the audience. So if you have any questions, please go ahead.